All right, so let's get started. Welcome everyone to our today's Immune Zoom seminar. Thanks everyone to be with us again today during the weekend. And then also I want to really thank our today's speaker, Dr. Xiao Shen from Zhejiang University to actually really generously agree to accept our invitation and then give us the lecture today. So really my pleasure to welcome our today's speaker, Dr. Shen Xiao from Zhejiang University. Xiao currently is, I think, full professor and also chair in the Department of Physiology in Zhejiang University. So very brief introduction for Xiao. Xiao actually completed his MD training in Beijing Union Medical College in China. And then after that, he moved on to pursue his postdoc training in basic medical research at Emory University. And by 2008, Xiao joined the faculty of Department of Biomedical Sciences, Cedars-Sinai Medical Center and at Los Angeles in US. After about 7 years, a research program conducted in LA, in the end of 2015, Xiao actually decided to move back to China and join the faculty of School of Medicine at Zhejiang University. And then last year, the year before last year, 2022, Xiao was actually promoted to the chair of the Department of Physiology. Xiao's lab mainly focuses on how resident immune cells participate in the maintenance of organ function and homeostasis in the brain and kidney. His lab actually particularly interested in studying the subtypes and the anatomical distribution of resident immune cells in the kidney, central nervous system, and the cardiovascular system. And then to explore how they perform their unique functions to maintain the functional homeostasis of the tissue and organs. And his lab is also committed to understanding the factors that induce immune imbalance during the development of the disease in order to provide strategies for disease prevention and early intervention. So I think today, he's going to tell us about some of his work from his lab about resident macrophage related to the kidney physiology and the immune responses. So I think without further ado, thank you so much, Xiao, for sharing your science with us, and we are very much looking forward to your talk. Please go ahead.
Thanks, Dr. Wu's invitation and introduction. Very kind. I'll share my screen. Okay. So you can see me clearly?
Yes.
All right. It's my great pleasure to share our recent research in this wonderful platform. I'm gonna talk about what we recently learned about the roles of resident macrophage in kidney physiology. Almost every tissue has its own pool of resident immune cells. On one hand, the tissue cells will provide growth factors for the survival of the immune cells. On the other hand, the immune cells will secrete the growth factors for the survival of tissue cells. This mutual reliance is manifested in an in vitro co-culture system in which the macrophage and the fibroblast provide growth factors for each other. However, emerging evidence unveils that the relationship between the tissue cells and the resident immune cells is more complex in steady state. Tissue cells will secrete other factors to drive immune cells differentiation and activation patterns. Moreover, the environmental cues will provide additional signals to educate the resident immune cells for the purpose that the immune cells can exert tissue-specific functions to help maintain tissue homeostasis and health. We had one finding that in the kidney cortex, macrophage have a role in supporting sympathetic nerve distribution and functions. As we know, tissue macrophage can be subgrouped according to their ontogeny. In one extremity, the microglia, the macrophage in the brain, are exclusively derived from yolk sac in the early embryo stage, at least partly due to the formation of BB in the later stage, which prevent the entry of blood-borne cells. In the other extremity, most of the intestinal macrophage in adults are derived from bone marrow, about 90%. And embryo and bone marrow-derived macrophage coexist in most of other organs, although in different ratios. It's well known that the kidney macrophage homogeneously and highly express CX3CR1, even higher than the monocytes. So we made use of CXCR1 pre-ER mice to do the fate mapping. It showed that while circulating monocytes eventually will completely lose the label, some kidney macrophages will reserve the label for over 1 year. To remove this, excuse me. And the half-life of the bone marrow macrophage is about 1.5 months. And this macrophage, which maintain the label, is self-maintaining. The reliability of this fate mapping system was confirmed by microglia, as over 90% of microglia were still labeled after 1 year. These self-maintaining macrophages are derived from embryo, since when we check the newborn mice, they had a comparable number of self-maintaining macrophages to the mice labeled in adulthood when examined 3 months later. Next, we analyze the difference in transcriptons of the kidney macrophage of different ontogeny. All samples highly express macrophage signature genes. There were over 2,000 genes differentially expressed between the two subpopulations. Surprisingly, many genes related to the nervous system were upregulated in the self-maintaining macrophage, while the downregulated genes are associated with metabolism and material transporting. So we next took a close look at the nerves in the kidney. The kidney are distributed with 2 types of nerve fibers, the sympathetic nerves and the sensory nerves. Consistent with the previous report, the context is densely innervated by sympathetic nerves, and the pelvis had many sensory fibers. The medulla had very few nerve fibers, so I do not show here. We find out that the self-maintaining macrophages, which is T tomato positive, in the cortex were in proximity to the sympathetic nerves, but not to the sensory nerves. When comparing the two populations of macrophage related to the sympathetic nerve, much more sympathetic self-maintaining macrophages, which are double positive for FO80 and T tomato, were in association with the sympathetic nerves over the bone marrow macrophage, which were single positive for FO80. 3D reconstruction showed that the self-maintaining macrophage were somewhat attached to the sympathetic nerves and sometimes wrapping around the nerve fibers. When we made the kidney live slices and observed their interplay for 2.5 hours, we noticed that the macrophage will extend and extract the processes along the nerve fibers. The extend, and this is the retract. In particular, we know that water deprivation will enhance activities of sympathetic nerves in the kidney. We found that water deprivation for 24 hours will increase the density of sympathetic nerve associated macrophage. and also expedited their protrusion movement, suggesting there was a need for a dynamic interplay between the sympathetic nerve and the nerve-associated macrophage. What sympathetic nerve do in the kidney? This is pretty much a physiology studies. During the long history of evolution, a common life-threatening situation is injury-induced bleeding, which will lead to hypovolemic hypotension. An immediate response to hypotension is to increase the sympathetic tonicity via the battle reflex. Sympathetic hyperactivity not only enhances heart pumping and vasoconstriction, but has multiple effects on kidney as well. It reduces the glomerulus filtrating rate, stimulates renin-angiotensin system, and directly increases water and electrolytes reabsorption by the tubules. The accumulating effect is a compensated increase of blood volume for saving the life. To understand the effects of the self-maintaining macrophage on sympathetic nerve in the kidney, we made a mouse line which expressed diphtheria toxin receptor. This receptor is under the control of 6CR1 after inducing. We waited for over 3 months. So by then, only self-maintaining macrophage expressed the DTR. Because the bone marrow-derived macrophage had been completely fleshly replaced, they lose the DTR expression. Then we gave a subcapsular injection of DT so that the drug had an effect only on the kidney, but not other organs. Initially, using the liposome-coordinated system, we noticed that the kidney macrophage will recover in 14 days after depletion. So we analyzed the kidney functions on day 14 post the PT infusion. It showed that after depletion of self-maintaining macrophage, the glomerulus filtrating rate increased, as well as the urine output. This is accompanied with an increase of excretion of many electrolytes, suggesting this is a solute diuresis rather than a water diuresis, fitting into the phenotype of sympathetic nerve dysfunction. Also, it showed that after depletion of self-maintaining macrophage, the sympathetic neurotransmitter norepinephrine was reduced in the kidney, as well as a decreased distribution of presynaptic marker. Therefore, we conclude that the self-maintaining macrophage are important in maintaining the renal sympathetic innervation. Excessive salt excretion can be easily compensated by an increase of intaking salt from food. We next put a low salt stress to the mice by giving them low salt diet. Some of the mice were supplemented with DOPS, a reagent which could be converted to norepinephrine in vivo as a rescue experiment. You can see that normal mice would reduce urine excretion during low salt diet to save the salts. But the self-maintaining macrophage deficient mice still had a boosted urine output, as well as salt over excretion. Eventually, while control mice gained a little weight in the 14-day challenge, the mice without self-maintaining macrophage had a weight loss, which indicates a loss of homeostasis. On the other hand, supplementing norepinephrine could rescue the phenotype. This finding unravels an irreplaceable role of self-maintaining macrophage in facilitating the sympathetic nerve distribution and activities in the kidney. Actually, the sympathetic nerve associated macrophage have also been found in adipose tissue. We are not the first to publish this kind of finding. However, the effects of those macrophages are to downplay sympathetic activities in adipose tissue. which is in the opposite to what we found that the sympathetic nerve associated macrophage in the kidney, which actually is helping the sympathetic nerve innervation. So I'll switch to another topic. The other finding came from our studying the medulla macrophage. The first finding is about the cortex macrophage. It's also talking about the specific macrophage derived from the embryo. Here we are looking at the medulla macrophage and found some interesting finding in steady state. So we know during the urine passage through the kidney tubule system, a significant amount of aggregated particles derived from various sources would form. For instance, 99% of the water in the glomerulus filtrate is reabsorbed, which leads to supersaturation of some solutes in the caudal segment of tubules. Second, apoptotic tubule epithelial cells routinely slough into the urine. In addition, some tubule segments will secrete a protein called uromodulin, which is a major component of protein cast. Above that, diet and certain diseases such as multiple myeloma also have effects on urine particle formation. So when we examine the global presence of macrophage in the kidney, what is the most striking finding is that we noticed that the medulla had the highest density of macrophage than cortex and papilla in both F480 staining and 6CR1 reporter mice. What are they doing is our next question. Interestingly, most macrophage in the medulla took a residence on the tubules with a high resolution reviewed that this is soma attached, very close attached phenotype. This macrophage also had a morphology like a microglia, in that they had multiple long protrusions, which was not observed in the cortex macrophage and other tissue macrophage, a very unique phenotype. Unexpectedly, a confocal analysis of thicker sections of the medulla with a high resolution microscopy revealed that many of the protrusions derived from macrophage actually penetrated through the tubules. When we removed the macrophage components in the picture, there was a hole on the affected tubule. PNA is peanut agglutinin, which binds to the luminal side of the collecting ducts. Collecting ducts is the caudal segment of kidney tubules. The staining of PNA further confirmed that macrophage protrusions were actually exposed to the lumen. 3D reconstruction showed that when wrapping around the tubules with their protrusions, most juxtatubular macrophage actually embedded a portion of their protrusion arms into the epithelium instead of directly inserting the protrusion tips into the epithelium barrier. The intraluminal 3D rendering of medulla collecting ducts exhibit the protrusions segments densely inlaying the luminal wall of tubules. Okay. This finding is intriguing because collecting ducts have the highest capacity with the lowest permeability among tubule segments, which will not allow the urine contents to affect the interstitium of the kidney. So to understand how macrophage penetrates through it, we co-stained with a tight junction marker 0.1. This displayed a strong labeling of the epithelial cell borders. It revealed that each trans-epithelial protrusion in most cases wrapped around more than one epithelial cells. The protrusion is very long. If the trans-epithelial segment of a protrusion cross over the border between two epithelial cells, it embedded into epithelium exclusively through epithelial cell body and circumvented the border without disrupting the overall junctional architecture, implying the minimal damage to the sealing capacity of tubular epithelium. This transcellular route was confirmed by electron microscopy of serial sections you can see the protrusions actually pass through the cell body, not through the border. Next, we use the two-photon microscopy to study the dynamics between the juxtatubular macrophage and the tubular epithelium. During the imaging sessions, although the cell bodies of most juxtatubular macrophage remained immobile on epithelial monolayer, their transepithelial protrusions displayed a dynamic dynamic extension and retraction movements appear in sampling the urine contents. To verify that the macrophage can intake urine contents, we intravenously injected a Fitch labeled sinistrin, which is a small molecule that can freely pass through the glomerulus. In 30 minutes, sinistrin arrived in the collecting ducts. By 60 minutes, macrophage had taken up Fitch sinistrin. But the larger particles are more concerning to the kidney tubules. To mimic the larger particles, we inject latex beads with a diameter of 2 micrometer via intrapelvic pathway. So some beads will be backflushed into the collecting ducts. So the beads cannot pass through the glomerulus. So we had to go through the pelvis. In 2 hours, you can see that some intratubular beads had been associated with macrophage protrusions. By 12 hours, some beads had been swallowed and transported to the macrophage soma, which is located outside of the tubule. Some macrophage had taken up multiple beads. This is very amazing. To understand the roles of macrophage in cleaning up tubules, we use the liposome collagenate to deplete the macrophage and then inject the beads into the tubules. After 36 hours, while the control mice were almost clean of beads, there were significant amount of beads still present in the medulla of mice deficient of macrophage. This experiment exhibited that urine flush alone is not enough to keep the tubule unobstructed, and the macrophage are required to facilitate the cleaning in a timely manner. However, the result of this experiment also raised a question, which is how the beads were eventually removed by the macrophage from the kidney. If the beads only be phagocytosed and transported to interstitium, the beads should be still stay in the interstitium. But actually, after 36 hours, the beads almost gone from the kidney. So we hypothesize that if the particles intaken by the macrophage are degradable by the cell, they will degrade them in situ. If the particles are indegradable, such as latex beads, juxtatubular macrophage can transmigrate to the lumen and escort the particles out of the kidney through the urine. This is a risky hypothesis. We got first to look for any sign of macrophage in the urine. When we examine the pee out of the bladder, we could not see macrophage, which is actually conceivable since the environments of urine are not livable for any cells. And after you pee the urine out, the macrophage already stay in the urine for hours. To circumvent that, we investigate relatively fresh urine by collecting urine from the kidney pelvis directly. Via this strategy, we were able to see macrophage in the urine of normal mice. This is in steady state. Interestingly, in steady state, the urine macrophage also contain the epithelium-derived components, such as AQP2, beta-catenin, suggesting that they had phagocytosed the sloughed epithelial cells. When we inject the beads into the tubules, we could actually detect the bead-associated macrophage in the urine. Some macrophage had taken up multiple beads. A direct evidence of macrophage transmigration to tubule lumen was derived from the two-photon microscopy. The video shows that a macrophage first resided in the interstitium between two tubules, and then it squeezed into the transepithelial tunnel made by its protrusion and gradually appear in the lumen of the tubule at the left side. The kidney stone is a disease reflecting an accumulation of intratubular sediments, which eventually block the tubules. The most prevalent kidney stone in human is oxalate calcium. We then treated the mice with sodium oxalate to induce an acute stone model. This strategy led to in 24 hours, a rapid and abundant calcium oxalate crystal depositions, exclusively luminal. And most of the crystals were in the medulla, not in the cortex. The medulla is the downside part of the tubule system. By 4 hours, we could observe an increased transepithelial behaviors of juxtatubular macrophage. By 24 hours, some intratubular crystals had been circulated by transmigrated macrophage. Actually, this phenomena was observed decades ago in human disease samples. Human intratubular casts always can be found as phagocytes on besides of them. But by then, it was not clear where these phagocytes come from. To investigate the role of macrophage in the progression of kidney stone disease, we induced a chronic oxalate stone model, which would lead to a mild stone formation in 7 days in normal mice. We specifically depleted the kidney macrophage by intravascularly delivering DT to the DIDTR mice. This is the first time it's reported that intravascular infusion of the drug can affect the kidney. Actually, it's very effective. Kidney stone formation was remarkably accelerated after macrophage depletion. Moreover, elevated inflation occurred in the mice depleted with macrophage in oxalate stone model, manifested by an increased neutrophil infiltration and upgraded generation of pro-inflammatory chemokines and cytokines. This observation underscored the role of macrophage as specific guardians, precluding both crystal deposition and possible damages inflicted by secondary inflammation, which is harmful to normal tissues. The transepithelial behavior is more prominent in the medullary macrophage than the cortex macrophage. They have a position difference. To probe the molecules involved in transepithelial behaviors, we next adopted a transcriptome analysis to compare the medulla and the cortex macrophages. In the gene ontology analysis, the top emerging term favoring medulla macrophage was positive regulation of cell adhesion, which is consistent with the outstanding and transepithelial behaviors of medulla macrophage. The interactions between immune cells and epithelium mostly occur during barrier inflammation, and these 5 molecules have been reported to mediate interactions in other organs in inflammation. So we found that among the 56 differentially expressed genes related to the top term, half are relevant to integrin beta-1 in the literature. Indeed, the medullary macrophage were relatively highly expressed the beta-1 in relative to the cortex macrophage. Strikingly, after treating white mice with a beta-1 blocking antibody, a medullary macrophage could be detached from tubules. At the same time, their sampling movement and the protrusion number were both decreased. More kidney stone will be formed when beta-1 was blocked in the oxalate challenge. The beta-1 forms dimer with integrin alpha. We ranked all integrin alphas in medullary macrophage based on their expression level and found the top 2 are alpha 9 and alpha 1, alpha 9 and alpha 5. Interestingly, alpha 9 beta 1, alpha 5 beta 1, these 2 dimers have one common ligand, which is fibronectin. Fibronectin is an extracellular matrix protein. Histology study revealed that fibronectin almost cover 100% of basement membrane side of medullary tubules, while only 10% of abluminal side of cortex tubules had the fibronectin deposition. In contrast, a well-known base membrane component, laminin, has 100% coverage on both cortex and medullary tubules. Actually, blocking the beta-1 fibronectin interactions by a peptide called the CS1 peptide could recapitulate the phenotype when beta-1 was blocked by its antibody. You can see a detachment of medullary macrophage from tubules and reduced protrusion numbers. Besides fibronectin, there is another common ligand for alpha-9 beta-1 and alpha-5 beta-1, which is osteopoietin. In naive mice, osteopoietin is only expressed in the outer medulla, but not in the inner medulla. Kidney stone formation by oxalate treatment could upregulate the osteopoietin expression by tubular epithelium in both inner and outer medulla, especially on the epithelial cells. SV peptide is known to block the ligation between beta-1 and osteopoietin. In resting state, blocking the ligation had minimal effects on the macrophage transepithelial protrusions. However, in oxalate stone model, previously we showed that it will increase the transepithelial behavior of macrophage. Blocking osteopoietin in this kidney stone formation could dampen an escalated transepithelial behavior, and so that the kidney stone formation was accelerated. So in conclusion, ligation with fibronectin through the integrin beta-1 is important for formation of transepithelial protrusions by macrophage in steady state. But in diseases such as kidney stone, the upregulated osteopoietin by epithelial cells would further enhance the transepithelial behaviors. So this kind of transepithelial behavior actually are translated by both macrophage and by the environments and the tissue cells. Finally, we studied pathological changes in the kidney in a mouse line deficient of kidney macrophages. Others previously showed that 6CR1 knockout mice had a remarkably reduced number of kidney macrophages, since 6CR1 has been shown to be important in the establishment of macrophage in both embryo and in adulthood. Compared to the same age wild-type mice, a 6-month-old macrophage deficient mice had already shown an increased deposition of intratubular protein, which were positive in all of HE, periodic acid SCIF staining and Manson staining in serial sections. With the age being advancing to 12 months old, more protein deposition as well as mineral and lipid depositions was apparent in the macrophage deficient mice. Here, we already excluded the possibility of an elevated concentration of these components in their blood, and altered the glomerular filtrating rates, and altered the reabsorption ability in the kidney tubules. So we can conclude that these changes came from the underrepresentation of kidney macrophage. To supplementing this phenotype finding by wild-type mice, we used wild-type mice, but weekly treated them with liposome collagenate for 3 months. The protein, mineral, and the lipid deposition were all increased in the macrophage depleted mice compared to the controls, especially since the liposome collagenate itself is a kind of lipid. So lipid sediments were particularly high in amounts in this model. So in conclusion, in steady state, organ tissue homeostasis was maintained by many means. The well-known mechanisms including neural and humoral regulations, which pass through negative feedback circuits. In the kidney, we recognize that red macrophage can also maintain renal physiology locally. They can prevent the disturbance at the first place. Of course, it's well known that they can also participate in resolving the consequences of disease so as to bring the homeostasis back. In the end, I will thank my teammates, especially Dr. Shi Peng and our funding resources. Dr. Zhu Qian significantly contributed to the sympathetic nerve project. Dr. He Jian is constructive in investigating the roles of medulla macrophage. They both help establish many means to study kidney macrophage. Thank you for your listening.